Okay, so I think this is where we left off in class. So epigenetics. Epigenetics refers to DNA changes that do not involve changes to the actual sequence of DNA. You know, we talked about mutations and, you know, how that changes the sequence of DNA, but these are changes to more the structure of DNA. You know, I talked a little bit about how DNA is packaged inside of a cell, and, you know, epigenetics really kind of refers to that. Now, these are really important changes, and a lot of them are completely normal. Okay, they act to turn on or turn off genes. They're important for example, inactivating one of the X chromosomes for the female. You know, I talked about that in class, how females inactivate one of the X chromosomes. It also causes the, you know, differentiation of stem cells. You know, this is how you can start by just, you know, a little 2-cell ball, and you form all these different cell lines from these stem cells. So epigenetics is really, really important. And these changes can occur at the level of transcription. Most of them occur at the level of transcription or at the level of, you know, translation. However, you know, if these epigenetic mechanisms are altered, they can certainly lead to disease. Okay, so DNA methylation. This is an epigenetic mechanism in which there is a methyl group, the CH3, chemical methyl group, attached to a cytosine base, and that's done by an enzyme called methyltransferase. And the key here is that methylation typically acts to repress gene expression. Another way to say that is it acts to turn off genes. Okay. Here's just an example. Some of our genes are known as tumor suppressor genes. They are genes that act to, you know, suppress the growth of tumors. And what happens is if you methylate a tumor suppressor gene, you're turning off the suppression of you know, tumor growth, and that can actually lead to things like cancer. You know, I always talk about smoking, and smoking is famous for causing excessive methylation to DNA, but also things like diet can affect these things. And we're also learning that, you know, these epigenetic things, you know, these changes to the DNA but not the actual sequence, can actually be passed down from generation to generation. And I think that's really profound. Methylation is also important for inactivating that X chromosome. And like I said, you know, abnormal methylation can cause things like cancer. Like I said, methylation of tumor suppressor genes. You can also have modifications to histones. For example, you can add or remove these chemical groups called acetyl groups, you know, acetylation or deacetylation from the histone proteins themselves. And this kind of changes how that DNA is packed, you know, around those histone proteins. So, for example, you know, since histones act to compact DNA molecules, which you talked about earlier, these epigenetic modifications can either upregulate. So if you add an acetyl group, they can upregulate or turn on genes, or downregulate if you take these acetyl groups off of the histone proteins. So I actually took this image. I told you about my one class I took that was just about transcription, so I stole this picture from that class. I thought it was really good. It's actually from Nature Review of Molecular Cell Biology. But anyway, you can see these ACs. These are the acetyl groups. When you add them to these histones, okay, these are the histones here. When you add them to the histones, it kind of opens up the DNA. And when that DNA is opened up, you know, our molecular machines can get in there and actually read the DNA and turn genes on. When you remove these acetyl groups, which is what's happening here, you know, these histones kind of pack all together, and that turns off gene expression. So genomic imprinting, you know, like I said, we typically inherit 2 copies of genes, one from mother, one from father. However, if one of the copies is what's known as imprinted, it is silenced and the other one is solely expressed. So basically, you know, some genes, a few genes, are expressed in what's known as a parent of origin specific manner. All right, and there's, I don't know, probably 75, 80 genes that are imprinted. Meaning that, yeah, you might receive a copy from mom or receive a copy from dad, but maybe only mom's is active, or maybe only dad's is active. And this occurs through things like methylation, through even deletions of genes or those histone modifications. So these really these genes are imprinted typically by these epigenetic mechanisms. Okay, so it's pretty interesting. You know, you always receive 2 copies, but maybe only one is actually turned on. So epigenetics in cancer, this is a huge booming area of research, but epigenetic changes in sporadic cancers are really common. Like I said, methylation of DNA repair genes, or genes that control the cell cycle, can certainly lead to cancer. Cancer takes a lot of hits. Just normal everyday cancer takes a lot of hits in the DNA before it actually develops. So it's not usually just one thing that happens and then you get cancer. It's a number of things. But like I said, smoking, that's been shown to cause epigenetic changes to the DNA, like methylation. And like I said, this is really important if a woman's pregnant and she's smoking, because this can cause epigenetic changes to the DNA of the fetus as well, and that can cause problems, down the road into future generations. So the study of epigenetics is really, it's booming right now, and it's very interesting, and learning more every day. So this is just a little cartoon showing kind of how all of these kind of play together. So epigenetics can cause instability in the genome because we're changing how it's kind of structured, and just how all of these come together to cause things like cancer. Like I said, cancer is typically a very complex disease. It takes a lot of hits in the DNA before you actually develop cancer. So it's just kind of showing these things. Okay, so some of these genetic and developmental diseases, starting with trisomy 21, also known as Down syndrome. So here there are 3 copies of the 21st chromosomes. Most of these are due to non-disjunction, which we talked about in the last video. There is a small percentage of them that occur by what's known as a translocation. I think I talked about that as well, where portions of chromosomes kind of swap. You still have, technically 3 21st chromosomes, but that third is actually attached to another chromosome. So 75% of these embryos that have 3 21st chromosomes, 75% will result in spontaneous abortion. 20% will die before age 10, and if you live past age 10, there's a life expectancy of about 6 years, so it's decreased life expectancy. So as far as some of the manifestations, there's always intellectual disability to some degree, low nasal bridge, hypotonia, a lot of times protruding tongue, low set ears. Males are going to be sterile, and females sometimes can reproduce, but certainly not as much as somebody without trisomy 21. So some other things here, there's increased heart defects, dementia later in age, immunosuppression. There's a whole lot of other things that go along with this. And you would think it's just another 21st chromosome. You wouldn't think it would have such drastic effects, but it does, because there's too much dose of those genes on that 21st chromosome. So determination of sex, I don't know why, I should have put this slide a bit earlier, but basically begins during the 6th week of embryonic development, and it's pretty much just determined by the absence or presence of the Y chromosome. If the Y chromosome is there, the gonads are going to develop into testes. If the Y chromosome is not, the primordial gonads will develop into ovaries. So if the Y is there, you're going to be male. If it's not, you're going to be a female. So the number of X chromosomes is irrelevant if the Y is present. I think I talked about that in class, because the person will still be phenotypically male. I mean, XXY, this is actually a condition known as Klinefelter syndrome, and they're still considered male, but they will have some kind of feminization of male traits. And like I said, females undergo X inactivation. So even if a female, has 3 X's or 4 X's, the 2 or 3 X's will actually be inactivated. So you only have one that's active. Again, down here you can see XX female, XY is male. So that little Y chromosome, that's what determines sex, basically. Okay, Turner syndrome. So Turner syndrome is also known as X monosomy. So this is a monosomy of the X chromosome. And this, again, is typically due to non-disjunction, and it's obviously present only in females. And only a small percentage of these conceptions actually make it to term. Okay, so 0.5%. So Turner syndrome, I should mention earlier I talked about monosomy. it's not compatible with life with one exception. I consider this the exception. So yeah, X monosomy. But anyway, patients, short stature, neck webbing. Neck webbing is kind of a key term here. They have, congenital defects of the heart and the kidneys. They tend to be shorter. Their intellectual ability is typically normal. Sometimes they have behavioral issues, but as far as the intellect, it's typically normal. Other comorbidities, you'll see a lot of diabetes, you'll see a lot of heart problems, and they typically have a shorter life expectancy. So on the left here, you can see some of the characteristics associated with Down syndrome. This is Turner syndrome. You can see the neck webbing here and some other things. So Huntington's disease, also, it's not always called this, but Huntington's chorea or chorea, just, to refer to Huntington's disease. But this is a progressive fatal genetic disorder. It's inherited in an autosomal dominant pattern, and it causes, movement problems, loss in cognition, and just neurodegeneration that eventually will kill the patient. As far as the autosomal dominant genetic disease, it is actually on chromosome 4. What happens is you get too many repeats in these Huntington's genes, and basically you have degeneration of areas of the brain called the basal nuclei, which are important for movement and motor function, as well as the cerebral cortex, which of course is important for cognition and a number of other things. Why Huntington's disease is particularly devastating is because it typically presents between ages 35 and 45, which is a lot of times after parents have had children. So if they start, maybe it's a spontaneous mutation, and they had a child at age 30, and then at age 35 they start showing these signs of Huntington's disease. That's devastating because there's a 50% chance that their children are affected. But manifestations, involuntary, and these fragmented movements, that's what chorea is, and that's why sometimes it's called Huntington's chorea or just chorea for short. And, like I said, progressive decline of cognitive functions, mood changes, it's a really, really difficult disease. Life expectancy, 15 to 20 years after the onset of clinical symptoms. That can vary a bit from that 15 to 20, it just depends. Typically, as this is passed down through the generations, it gets more and more severe. Okay, sickle cell anemia. This is a autosomal recessive disease, and this affects red blood cells, specifically hemoglobin. So what happens is you get normal hemoglobin that turns into what's known as sickle hemoglobin, or HbS, in the red blood cells. And this is caused by a missense mutation. Okay, missense mutation, which I talked about earlier. So remember, in a missense mutation, you have a change in the amino acid. And I believe that in sickle cell, it's a change from glutamic acid to valine. I think that's what it is. But it's just it's a single base change that causes a change in the amino acid sequence, and it has these devastating effects. So what happens is these red blood cells become misshapen, they become sickle-shaped, they become sticky, they become fragile, they clog up small blood vessels like capillaries, and yeah, it's going to be devastating. So what happens is, there's lots of pain. There is, susceptibility to infection because you have all this congestion in the spleen. The spleen is trying to get rid of all of these sickled cells, but it really can't keep up. Sensitivity to cold, okay, because of the occlusion of those small vessels. Hypoxia of the tissues, obviously the anemia, because for one, you have these red blood cells that can't really carry oxygen as effectively, and then they are kind of clogging up these small vessels, so you can't get the good red blood cells to the site. So it's kind of a double whammy when it comes to the anemia, and also jaundice, especially during a sickle crisis. So you can see this is a normal red blood cell. A normal red blood cell is nice and flexible. It's just kind of like a bag of hemoglobin. And then these are the sickled cells, which obviously are very misshapen. They get caught in the small vessels, and they can occlude blood flow, and that leads to the hypoxia and anemia and the pain, and can be really, really devastating. Neural tube defects. These are developmental disorders, and basically you have incomplete closure of the neural tube. So during embryonic development, you have a structure known as the neural tube, which will kind of like close. Well, it's supposed to close, and what happens when it doesn't close is you can get some pretty significant manifestations. So spina bifida, this is when you have incomplete closure of the vertebrae, and there are three main types. There is spina bifida occulta. This is the least severe. Okay, a lot of times people don't even know they have it. You have meningocele. This is the second most severe. This is when you have a herniation of the meninges through this incomplete closure. And then the most severe is myelomeningocele. So in a myelomeningocele, you have a herniation of the meninges plus the spinal cord, or the spinal nerve roots. And this is the one that's associated with, things like paralysis. Anencephaly, this is when you have incomplete closure of the skull, and this is not compatible with life, at least not for very long, for obvious reasons. So here is showing, a normal, so this is kind of a transverse view here, and you can see, this is normal, vertebrae, the spinal cord is completely enclosed like it should be. In spina bifida occulta, you have a small defect in what's known as the posterior vertebral arch. A lot of times this causes no problem. Some people don't even know it's present. Sometimes these patients will have like a tuft of hair at the small of their back. Meningocele, this is when you have a herniation of the meninges through this defect of the posterior vertebral arch. The spinal cord, though, is completely intact and is in place. This can be repaired, and a lot of people will not have any ill effects from this. The most severe type of spina bifida, though, is the myelomeningocele, when you have a herniation of the meninges plus the spinal cord or the spinal roots. And this is the one that is associated with paralysis. Okay. Anencephaly, okay, there's no closure of the skull, the brain is exposed, and this is not compatible with life.